It turns out that the allele frequency is the probability that a neutral allele will become fixed eventually. What? So in this figure here, the allele frequencies all started off at 0.5, like we said. And indeed, there's a 50% chance that the A1 allele will become fixed in the population. And that's what happened in these models. About 50% of them reached fixation for the A1 allele, and about 50% of them reached fixation for the A2 allele, both of which, again, started at a 50% frequency. It's equal to the probability that they would eventually become fixed in a population. Okay, same thing. Well, it turns out that the probability of a brand new mutant neutral allele is equal to 1 over 2n. Now notice, this is not 1/2. This is not 1/2 of n. No, this is very different. This is 1 over 2n. So we're multiplying 2 times n. N again is the population size. So how does this make sense? A brand new neutral allele. It just popped into existence. It has no phenotypic expression. And it's in a population of, let's say, 4 individuals. 4 diploid individuals, let's say. Well, each diploid individual has 2 alleles. And so the total number of alleles is 8. This brand new mutant neutral allele, it's brand new, so there's just one of them. So its frequency is 1 over the total alleles, 1 over 8. Okay. If the population was much bigger, which it probably will be, that was a silly number to pick. Usually population sizes are in the hundreds or thousands or millions. A brand new allele just popping into existence in a massive population of thousands of individuals, it's going to have a very low frequency and therefore a very low probability of reaching fixation eventually. It's possible, though. Okay. If it is going to reach fixation, if that brand new mutant allele, which is neutral, if it is going to reach fixation, it turns out that it takes exactly 4n number of generations to reach fixation. So in a smaller population like the one down here in B, it's a smaller population just because this y-axis is smaller than this one up here. The y-axis is much bigger. I'm assuming that's more individuals. This one down here in a small population, it only took this amount of time, right? This amount of time to become fixed in the population. But that same allele in a large population, it took this amount of time, right? So we're talking a pretty big difference. In a larger population, it will take a longer amount of time for that allele to drift through all of the individuals, right? Generation by generation. And that's what we're talking about down here, by the way. This time is by generation. Generation 1, 2, generation 3, generation 4, generation 5, for example. It's going to take a brand new neutral allele a lot longer to drift to fixation when there are more individuals in the population. Kind of makes sense. So the neutral theory says that the average neutral allele fixation rate for a population is independent of population size. So that kind of is counterintuitive based on what I just said. It takes an allele longer to reach fixation in a larger population. But since it's a larger population, there are more chances for mutations. There are more individuals who might be getting, say, some brand new mutated neutral allele. So in other words, a larger population has more alleles that could mutate, but it takes each one a longer time to drift to fixation. Or, from the smaller population's perspective, a smaller population has fewer alleles available to mutate, fewer individuals, but they drift to fixation faster, and therefore a small and a large population size will be accumulating these neutral alleles that are becoming fixed. They'll be accumulating them at the exact same rate over time. So this is really important for calibrating molecular clocks and doing things like building cladograms that are based on kind of divergence times. Okay. Heterozygosity kind of has to do with what we're talking about here. Heterozygosity is the proportion of genotypes in a population that are heterozygotes for some locus. It's also a measure of genetic variation in a population. According to the neutral theory, the amount of genetic variation in a population should be proportional to the population size. All right. Drift simulation. Oh, yeah, I already had this slide up here. Remember this from earlier. This was just showing the fate of a single allele, right? Regardless of its fate, this population, say like right here, this one model population in red, or now in green, it is entirely homozygous at this stage right here, at this locus, right? Allele A1 has been completely lost, and so all individuals have the A2 allele. They've lost heterozygosity, in other words. So every time an allele does become fixed, even a neutral allele, every time it becomes fixed, that population has just lost a little bit of heterozygosity. All of the individuals will be homozygous at that locus when an allele becomes fixed. So you notice that the loss of heterozygosity due to genetic drift is showing a similar pattern based on population size as those genetic drift models that I just had on that last slide. So here are the same population sizes as the last slide. N equals 4, here's N equals 40, and here's N equals 400, and much bigger. And due to genetic drift, smaller populations will lose heterozygosity much quicker on average than larger populations. They'll lose heterozygosity much slower. Even larger populations will be losing heterozygosity much, much slower. This is based on population size. So it turns out that you can calculate the expected loss of heterozygosity in population. And the way to do this is 1 over 2 times n. Again, this is not 1/2 of n, this is 1 over 2 times n. So from generation to generation, this is the amount of heterozygosity that's expected to be lost in a population. 1 minus that, 1 minus 1 over 2n, is how much heterozygosity would be expected to be retained in a population. So notice that this equation is clearly based on population size, since we've got the population size n right there in the denominator. So just kind of doing a quick review of what 1 over 2n actually means. It kind of means a few different things here. 1 over 2n is the frequency of any new mutant allele. I explained that earlier. It's also the probability of any one allele becoming fixated in a population eventually, if it's brand new and it just popped into existence. 1 over 2n is also the average proportion of heterozygosity lost per generation by drift. That's what I was just talking about. Okay, 1 minus that, 1 minus 1 over 2n is the average proportion of heterozygosity retained per generation. And then this is one from earlier. 4n is the average number of generations it takes for a new neutral allele to become fixed in a population, if it's going to. Okay, so if you wanted to kind of figure out what would be the expected heterozygosity in the next generation, or in 10 generations from now, you would use this equation down here. You would need to have some information now, the current heterozygosity, for example, and the population size. If you've got those two values, then you can punch them into this equation, your heterozygosity now, plus that n for your population size, and you can figure out what the heterozygosity will be in the next generation. This is just another way to say it, the G plus 1. You often see that in textbooks and stuff. G plus 1 just means the next generation. G means the generation now. And so if you wanted to know in 10 generations, you would do the same equation, but to, say, the 10th power. Okay. So now I wanted to talk about nucleotide changes in codons. Here is a nice amino acid chart. You did something on your discussion assignment about replacement and silent sites, talking about replacement distances. So let's talk about this in light of, say, some codon. Pick a codon, any codon. All right, let's see. Did you pick GGG for glycine? Good choice. That's what I picked too. In this amino acid, glycine, down here. The codon GGG, the one I picked, notice that if there was a mutation in that very first codon position, right here, that first nucleotide position, say it was CGG or TGG or anything, then we would have to go to a different box. We'd have to go up here, the AGG or CGG. And either way, we would end up with a different amino acid. So that first site is a replacement site. The second site is the same thing. If there was a mutation here, say a GAG or a GCG or a GTG, well, we would again end up with a different amino acid, and that's why the second position is also a replacement site. The third site here, though, for glycine at least, this third position is a silent site. Because if there was a mutation, say to an A, or to a C, or to a T, it would end up with the exact same amino acid. So it wouldn't change anything drastically that's being expressed. You'd have the same amino acid, and so we call that a silent site. Or in other words, a synonymous site, meaning the amino acid would be synonymous, it'd be the same one, unchanged. So for amino acid replacements, most new mutants have very low fitness and are removed by selection. Imagine some random mutation, it's probably going to be deleterious, odds are, and removed from selection, never really seen in a population much. A very few number of changes will confer high fitness and will be favored by selection and will increase in frequency. But the vast majority are actually nearly neutral and accumulate approximately by the neutral theory prediction. So actually a lot of even these replacement sites can end up kind of having neutral effects. We talked about the molecular clock very briefly earlier this semester in light of building cladograms with the UPGMA method. One requirement for that was that the data was ultrametric or clock-like. That was the ideal model. Or in other words, the say genetic distance from A to C is the same as the genetic distance from B to C. This works well, or it really works only, rather, with neutral changes over long periods of time. So in other words, those silent changes, remember, those are the ones that are effectively neutral. They're not changing anything that's being expressed that would result in some big deal change. So silent sites are ideal for sequencing if you're trying to use them to build cladograms that are ultrametric in their data. So neutral drift. It's predictable because it's random. It ends up making these clock-like changes that accumulate over time. Selection, on the other hand, it's highly variable. Genes that have been highly selected for or against are not generally the best types of alleles or genes to sequence in terms of building cladograms. Okay. But there's something about these codons, like I said, for this GGG, you would expect to see these mutations pretty frequently in the genome. You would expect to see these mutations accumulate over time because they mean nothing. And so randomly, they're going to be accumulating over time. You'd expect maybe an equal proportion of those 4 different types of codons that all code for that glycine. But what can happen sometimes is you end up with something like this. This is called codon bias. So say the molecular machinery gets more used to one of those codons more than the others. You can actually end up with these silent changes, or rather changes that are in silent sites that are not truly silent. So in other words, that third position in the nucleotide is not necessarily always neutral. I just had to say that this codon bias thing is important, and you always need to check for that codon bias before using the DR over DS method, which is what we're going to talk about now. This is the method for figuring out if the type of selection you're looking at is positive, neutral, or negative. So here are these 2 alleles. We can just assume maybe they're in 2 different species. And notice that a lot of the nucleotides are identical between these 2 alleles. Some of them are different, though. Well, like we figured out earlier, those first 2 positions in a codon are almost always replacement sites. that third position is generally a silent site. And that's the pattern that we see here. Replacement, replacement, silent. Replacement, replacement, silent. Replacement, replacement, silent, etc. Okay, so in order to figure out the silent and replacement distances here, we need to see where the differences are accumulating between these two alleles, or between these two species. So let's start with the replacement distance. We need to kind of account for all of the differences that we find between these two alleles that land on a replacement site. So it looks like there's one right here, and it looks like there's another one right here. And I think that might be it. So there are only 2 differences between these two alleles out of the total number of replacement sites. That is how we get the replacement distance, or in other words, dr. Okay, to get the silent distance, we need to account for all of the differences that we see between these two alleles that land on a silent site. So looking at this first codon, it looks like there's one, right? The third position, the T is different from the C. So there was one. Oh, same with this next codon. Oh, same with the next one and that one. So out of the 5 silent sites in these sequences, 4 of them are different between them. This is our silent distance. And to figure out if we've got positive, neutral, or negative selection, we need to do dr over ds. So in other words, 2/10 over 4/5, which I calculated to be 0.25, which is less than 1. So let's check out what that means here on the next slide. When your dr over ds is less than 1, like shown here, we've got negative selection. We would expect those gene regions to have a high conservation. Maybe these are very important functioning parts. Lots of important biosynthetic pathways are undergoing negative selection, where if there are changes that are accumulating over time, they're more so in the silent sites. When your dr over ds is equal to 1, or in other words, your dr is equal to your ds, then we call this neutral evolution. These are good ones for figuring out, for making a molecular clock, for, as they say, calibrating a molecular clock. Things like processed pseudogenes, microsatellite alleles, things that people often call junk DNA that have no phenotypic expression. Positive selection is when change is advantageous. So it's when you have more replacement site changes, which are big deal changes, accumulating over time. Just so you know, the textbook, they use dn over ds. That's for synonymous over non-synonymous. Same thing. Oops. Just did that. Okay, so which genes, say someone had a question. They wanted to ask which genes were most important during human brain development, right? Our brain is very unique relative to other species around the world. Pick something that's really unique in your favorite organism. You like birds. Well, peacock feathers are pretty stinking unique. And why are they so different? What genes were responsible for making those peacock feathers so darn special, so different? What genes were responsible for making our brains so special, so different from other such organisms, especially from other close relatives? Well, in order to figure out what genes might be responsible for those, we first need to sequence mRNAs from both human and non-human primate brains, so from some other close relative, say a chimp or something. So you're just sequencing genes. Or say in my peacock feather that I'm talking about now, you might sequence feather genes from peacocks and other close bird relatives, like a grouse or something. You align those coding sequences, and then you want to try to find out which of those genes have a high dr to ds ratio, or in other words, which of those genes are undergoing positive selection. Selection for that's increasing the amount of changes from generation to generation. That's benefiting, or that where there's an increase of fitness for individuals who have lots of changes that are resulting in a new amino acid, which is resulting in a potentially different function or different expression. Those would be genes where we would expect these differences to have been accumulating between, say, a feather that is highly ornate in one species and the feather that is not in another species. Or a brain that is huge in one species and much smaller in a closely related species. Positive selection is probably responsible for that. Okay, I think that is my last slide. I hope you enjoyed, and I'll see you next time.